Okay, this is going to be the fourth video for Chapter 8, obviously, Section 8.4, which is about individual differences and variability. We're continuing our discussion on individual differences in a between subjects design. The learning objectives 5 and 6 are what we're going to be talking about here. And to remind you of what those are, we're going to talk about the individual differences influencing variability within the groups, which I'll explain what that means in a minute, and then also how that influences the interpretation of the results, and what are the options for reducing or controlling this variability, within the groups, so that we can make sure that we are able to interpret our results accurately and get true results. So, the effects of variability and in between subjects design. So, large differences between individuals produce a large variance. So, variance may be a term that you're familiar with as far as like what it basically means, in just general terms, but as far as in the scientific field and statistics, you may not have heard that term or know what that means. So, it is the size of the differences between all of the scores. Okay, and so if all of the scores that the participants on your dependent variable, whatever you're measuring, if all of their scores are similar, or mostly similar, then there's going to be a small. You would say that there's a small variance, right? They don't vary a lot. The scores don't vary a lot. But if there's large differences between the scores, if they're kind of all over the place, then that would be a large variance. You would say they vary a lot. The scores vary a lot. So there's a large variance. And so to kind of give you an example of that, this table 8.1 has two simulated populations. And in population A, the differences between the scores are small, so it's a small variance. And in population B, it's a large variance. So I'm going to scroll through here and let you guys kind of see. You can't see population B yet, it's over here. But for population A, you can see that it's mostly in the high 30s, low 40s, right? Almost all the scores fit within that range. Now, if I scroll over and you see population B, you see that most of the scores, or that these scores vary a lot more. There's a larger variance, right? There's 20s all the way to 60s, right? That is a much larger variance. And so that kind of gives you a visual example of what I mean when I say variance. Your book gives the example of variance being kind of like radio static or cell phone interference, interfering with a radio or a cell phone signal. So because of variance, if there's a large variance, it can be more difficult to see a pattern in the data or to see the results of your experiment, to see if there's really a difference between the groups, if the treatment, if the independent variable had an effect on the dependent variable. It can be hard to see that if there's a large variance. Okay. So it creates that static, right? It interferes with the signal, basically, is what that example was, or that analogy is pointing to. So how do we increase or decrease variance? So this last part of the slide says that increasing or decreasing the variance, first of all, can have a significant influence on your interpretation of the results, which I kind of alluded to a second ago. And significant means that, like when you're talking about statistical significance in a study, it means that it is very unlikely that the difference would occur if there were not a consistent difference between the treatment conditions. So when you're saying it's statistically significant, you're saying that the results that you got are very unlikely to have occurred if there wasn't truly a difference here, if the independent variable wasn't truly making an impact. And so or you can just say that it's another way of saying it is that it would not have occurred by chance, right? That there is something actually happening. The independent variable is actually making a difference in the dependent variable. So I'm going to skip these next two slides. We're going to come back to them. I want to show you this figure 8.4, 8.3 and 8.4 from your textbook to kind of give you another visual example of what it is that we're talking about when we say it could impact the results. So here you can see this is the population A that we just talked about a second ago. So you can see the control condition and the treatment condition. You can see that the population or the scores on the dependent variable don't vary a whole lot, right? And the treatment condition, they went up. So the treatment had an effect. And you can very clearly see that here, right? Because the red, you can see at the bar graph, the red's much higher. If you look at the column information or the table there, you can see, again, you can visually see that the numbers, all the numbers go up. They're all higher, right? So it's easy to see. But that's because there was a small variance in the control condition. There's a small variance in the population or the sample to begin with, right? Now, if we go here, when the individual differences are large, so there's large differences, there's large variance, look how much different that in the treatment, the difference between the control condition and the treatment condition, this is the simulated thing is the same, but you cannot see the effect of the treatment here, right? I mean, if you look at that, you cannot see visually the difference there. Now, it is also important to show you visually, you can see it, but also statistically, this would not have been statistically significant. So you would not have seen that the treatment had an effect here. But here you would have, this would have been a statistically significant result. Okay, so that is why limiting the variance, these individual differences, is so important. So going back to these slides, large differences between the treatments are good. We want it between, because if there's a large difference in scores between the two groups, that suggests that the independent variable had an effect on the dependent variable. So we would love to see large differences between the groups. What we're talking about here is the differences within the groups, the individual differences between the participants within each group. We don't want that to be large. We don't want there to be a large variance within the groups, because again, that adds that static, that interference. Then we can't really determine whether or not our independent variable or our treatment had an effect. So the differences that exist inside those treatment conditions is determining that variance again. And so we want that to be as small as possible. The large variance is what hides those patterns in the data, like I just said. That's just kind of another way to word it. So what can we do? Well, researchers try to increase the differences between the treatments. Again, we're trying to maximize the impact the independent variable has on the dependent variable, or the treatment has on the scores. And then we try to decrease the variance within the treatments as much as possible. We try to limit those individual differences and that variance within the groups as much as possible. So an example they give is the color of a room impacting mood. So the way to increase the differences between the treatments or between the groups would be, you wouldn't want to have different shades of the same color and that be, like a light green and a dark green, and then say we want to see if color of the room affects mood. Well, two different shades of green is probably not going to show you an effect because they're so similar. So to maximize or increase the differences between the treatments, you would want like a muddy brown color and a bright yellow color, or something that has a larger contrast, right? And you could have more than two treatments if you wanted to, make sure you could do, different colors. But the point is that you want to make sure that the differences aren't subtle between your groups. You want to make sure that your independent variable influence is not subtle. Now, decreasing the variance within the treatments, in this example, how would we do that? Well, there's some there's four things that we can talk about in terms of minimizing the variance within the treatments. So in that experiment with the color of the room and the mood, you would want to make sure that you are standardizing the procedures and the treatment settings. So you want to make sure that the participants are treated the same within each group. So if they were all in the room together in the yellow room, you would want to make sure that you didn't say or do anything differently with one of the participants than you did with the other, especially something that could have affected their mood, right? Because then that's going to be a confounding variable that's going to influence the dependent variable of mood, and that had nothing to do with the color of the room. And so that is part of it, but also you want to make sure that you're treating them the same so that any individual differences that they may have had coming into the experiment, that those aren't made worse or amplified in any way. If you treat them all exactly the same and have the same procedure and everything is standardized, then you know for sure that you're not making those individual differences any worse. And so this is also really good. Standardizing procedures and treatment is also really good because it's a validity check for sure, but also it's good for other people to understand your research because it's very clear and structured. It's better for them to be able to understand what you did and why you did those things. And it's also great for replication purposes if somebody wants to replicate your experiment. And so it's good for minimizing variance, but it's also good for other reasons as well. Now, another way that you can minimize variance is by limiting the individual differences as much as possible, right? And so we kind of talked about that in 8.3 when we talked about restricting the range, like if we're talking about age or whatever. And so you want to limit those individual differences as much as possible, whether that's holding, variables constant or limiting that range. Now, that actually helps both types of variance. That actually helps the between group and the within groups. Like that helps increase the differences between the groups and minimize the variance within the groups by doing those, by limiting those individual differences. That helps both. So that's probably the best method here. However, with like everything else in research design, there's a give and take. It also limits your external validity again, because if you are limiting the range or holding variables constant so much that you can't generalize it to other age groups or other genders or other populations, then you're limiting your external validity, right? You can't generalize your results. And so that is a huge downside to that. So again, there's a balance that you have to try to find there. And in case you're thinking, well, then there's just no hope, there will be more discussed in chapter 11 about other designs and other ways to kind of mitigate this, how to help navigate this a little bit more. But it also, gets into more complicated procedures. So that is going to be later in the textbook. Now, random assignment and matching is on here because we talked about that in 8.3. This as a way to help the between groups differences and limiting those individual differences. But this does not work for variance within groups. So I wish the PowerPoint had this differently, but because it looks like random assignment and matching is an option for minimizing variance within the treatments, but it's not. It's on here to remind you that it is not that it does not work for within groups. Because if you think about it, matching and balancing those characteristics, those individual differences across the groups, yes, it helps the differences between the groups, but it doesn't help making sure that your within groups um, your within groups variance could still vary depending on what variable you're looking at. And so um, you have to be careful. Not that those aren't good procedures, because they are, but you just have to be careful. It depends on the goal of what your research is, right? Um, and so another way is sample size. Sample size, larger the sample, like in most cases, can be better, right? For most purposes. The larger your sample size, the less likely you're going to have these, some of these issues. But your textbook talks about the square root rule. What that means is that if you needed to increase, or I'm sorry, decrease the variance within the groups by a certain amount, you're going to have to, however, say it's you need to decrease the variance by a factor of 4. I think is what the book uses the example. The square root rule means that you would have to then increase your sample size by a factor of 16, because 4 is the square root of 16. So that would mean you would have to take the number of participants that you had and multiply it by 16. That's huge, right? That means a very large sample size. That is a very different sample size than what you started with. And so, again, sample size being larger helps, but sometimes that's not practical. And also, um, depending on how much variance there was to begin with, it may be very impractical, because you may not be able to have a large enough sample to even impact or make a difference on the variance within the treatments. So that's just kind of a quick overview. Of course, they go into more detail of all of this in your textbook. Now, this little discussion activity, I encourage you to think about this for a minute. You can pause this, you can screenshot this, but in a between subjects design, one group of participants represents a single treatment condition. One obvious way to help minimize variability within each group is to be sure that all participants are treated exactly the same. That was that standardization we just talked about. What are some ways that you could do that? What are some precautions that you could take to make sure that your groups are treated exactly the same? I'll give you an example. Writing out the instructions and having the researcher read them to them, instead of just giving it to them and letting them read it, because then you have to worry if they're actually comprehending what they're reading. Everybody has different reading comprehension. Everybody reads at a different speed. And so if you can read it to them, then you're making sure that they are not skipping parts of it, that everybody's hearing the same thing, and everybody is hearing all the instructions in case they skipped parts, like I said, or missed part of it or didn't understand part of it. Also, if you don't have it typed out, then they could end up saying something different to each of the participants. But having it typed out and having them read it standardizes it, makes sure they're saying the same thing to every single participant. So that's just one example, but there are other precautions and things that you can probably think of there. So lastly, for 8.4, we are going to do the learning check here. So which of the following maximizes the likelihood of a successful research result? So which one's maximizing the success here? So hopefully, from looking through this, you know that decreasing differences between treatments is never a good idea. In a between subjects design, we want those differences between the treatments to be large. We want the independent variable to have had an effect. So the two that start with decrease the differences between treatments obviously is incorrect. And then the other two, one is decreasing the variance within the treatments and one is increasing the variance. Hopefully from doing all of this, you know that you want to decrease, we want to minimize the variance within the treatments. So the answer is increasing the differences between so that we know the independent variable had an effect, and we want to decrease the variance within the groups so that, again, we can see clearly the difference between the groups. Which of the following is an option for limiting the variance within treatment conditions? So we're limiting the variance within. So what are the things that we can do for limiting variance? Can we hold a participant variable constant? Can we randomize the participant variable across treatments? Can we match across treatments? Well, as I mentioned before, randomizing and matching does not actually help within groups variance. That is for between the groups. So the only option here was holding the variable constant. If we want to limit those individual differences within the groups, you need to hold that variable constant. Or that is one of the options, is holding the variable constant. The other two are not. Which of the following is a potential problem with holding a participant variable constant? So we just talked about that. What's the problem with holding it constant? And hopefully you know from what we just talked about that it's starting the external validity. Because again, if you are holding that variable constant, then that means that you are limiting your sample, your participants. You are limiting either their gender, their age, their whatever the variable is. And so because of that, you will only be able to generalize your results to that small group of whatever characteristic it is. You won't be able to generalize it to a larger population. And so, again, it's give and take. It's something to think about when you're trying to figure out the best ways to design and structure your research. So that is it for 8.4. If you guys have any questions, of course, I'm here. And I will post the next video for 8.5. That will be the next one we do.